bench to bedside research is it's no longer unidirectional. In my lab, I have collaborators everywhere in every aspect of research. And I think I talk to them almost every day in order to make my research more timely and better. So we go from the diagnosis and treatment in first line and second line. This is typically where from the patients, it could be blood, it could be other samples, tissue, and other body fluids. And then we build cell lines and mouse models from these either blood or the tissues. And then we do array of techniques. And I'm going to talk about the utility of each because, there's a cost associated with all of this. So then we take that to what we call preclinical research. This preclinical research then informs the clinical trials. It could be phase 1, phase 2, phase by, several fantastic clinicians who will tell you a lot more about this phase 1, phase 2, and phase 3 trials. And then we learn. It's not unidirectional anymore. We learn from these trials, we go back to the lab, and then we do this process again to make the process more efficient, more better. That's always the goal of translational and basic science. So let's talk about, the cell lines and the mouse models. So, if you've been to conferences and talking about lung cancer, you will come through this slide a lot. How has cell lines and mouse models really helped us in developing better therapies, right? So I think non-small cell lung cancer is a true testament of molecular testing in terms of developing targets and better therapies, right? Now, when you think about lung cancer, it's no longer one disease. We can divide lung cancer into these different subtypes, which are driven by these driver oncogenes that you will hear about later in the day, like ALK, EGFR, KRAS, and there are drugs against it, which the patient gets when they come into the clinic. And now, thanks to basic research and translational research, we understand so much more about the disease that we are thinking beyond just oncogene, the single oncogene. We are thinking about co-mutations or the alterations in the genes that happen together. And one very good example is the KRAS mutation, along with its two co-alterations like STK11 and KEAP1. So this was a study that came out of Memorial Sloan Kettering Cancer Center, which looked at KRAS mutations and all the other co-mutations that predict poor outcome in this subtype of the disease. And what they found was that in a KRAS mutant tumor, having a co-alteration in these two genes, STK11 and KEAP1, actually predicted the worst survival and the worst prognosis in these patients. So we went back and we looked into these data sets of 1,235 metastatic lung adenocarcinoma cases, and we found indeed in these patients, if you just concentrate on the black line here, the patients who had KRAS mutation along with STK11 and KEAP1, they had a much worse prognosis than the patients who did not have mutations in these genes. So the real question that we asked in the lab is, what is the function of these genes and why do having these co-mutations make this cancer so aggressive? So for that, we use the cell lines in the lab, and that is the utility of cell lines. So we use these two cell lines, H358 and H292. We actually knocked out or we deleted these genes in these cell lines, and we wanted to study what is the biology, what can we learn from the RNA, from what I told you, what can we learn from these processes called RNA sequencing? I'll talk about RNA sequencing in a bit. And also with CRISPR screens, where we delete different genes in these models, and then we try to identify what happens and what are the phenotypes of these cell lines. And what that led to, I'm not going to go into the details, but what that led to is to understand that there is a specific vulnerability in these cancers, which is called a ferroptosis or a cell death pathway. And there are several genes that are involved in this process, which could be targeted. And, right now, there are preclinical studies and also translational studies going on in trying to understand the importance of ferroptosis in this cancer. So this tells us that we can learn a lot from cell lines, and this can actually be taken into the clinic.